So we're going to start with glycogen metabolism. So the first place we're actually going to start is with degradation. Specifically, we're going to be starting with digestive degradation. This is one of the interesting things about talking about starch and glycogen, which is that we can both make glycogen, degrade glycogen, but we also get starch and glycogen from our diets. So we're going to actually start with what happens when we eat this. So when we obtain starch and glycogen from our diet, what first starts on this is amylase. So that alpha amylase is going to be an endoglycosidase, and it's going to basically break things down in not exactly a random order, but in various chunks and units. So that alpha amylase is going to cleave things, but it's going to cleave things into disaccharides and trisaccharides, into maltose or maltose triose, other small oligosaccharides. So that amylase isn't very picky. It's just kind of nomming on different parts of a starch or glycogen molecule. It's present in saliva, as well as pancreatic secretions, and it specializes in hydrolyzes that alpha-1,4 linkage that's present in glycogen and starch. It is active on either side of the branch points, but its activity is really reduced near the branch points. So the closer you get to that alpha-1,6 linkage, the worse this thing is going to do. There are debranching enzymes then that can cleave those limit dextrins, and we'll take a closer look at that in a minute. But this debranching enzyme actually has two different activities that it has. One, it actually transfers trisaccharide groups, and it can actually cleave that remaining single glucose unit from the main chain. So it has two different activities. We call it a debranching enzyme. So that debranching enzyme, looking a little bit closer, so one, it's going to remove three of those sugar units and move them to the end of the chain. So this is known as a limit branch. So it's something that that limit branch is basically something that the amylase leaves because it's close enough to that alpha-1,6 linkage that the amylase can't really get to it. So since the amylase digestion leaves those highly branched limit dextrins, this debranching enzyme, which has two activities, is first going to move those three sugars to the end of the chain, which then amylase can keep going on. And that same debranching enzyme has a second activity, which is this alpha-1,6 glucosidase activity. And so that's going to go ahead and release that one residue. So the debranching enzyme has both the gluconotransferase activity as well as that alpha-1,6 glucosidase activity. So two different activities, one enzyme. And so by that way, it is going to release that one single glucose molecule from that branch point and then just move the other set. Whenever we're talking about glycogen, this is the one that gets even trickier. So in humans, it's a little bit easier to be like, starch from plants, we eat that. Glycogen, we make that, but realize that many humans are also omnivores, which means we eat other animals, which means that we will be breaking down both starch and glycogen, assuming a diet that has both plants and animals in it. So dietary breakdown of starch and dietary glycogen is unregulated. So those amylases just go to town on things. Those debranching enzymes just go. And almost 100% of that digested food is absorbed and then metabolized. But tissue glycogen is an important energy reservoir. So the glycogen that we make is going to be tightly regulated. So both its synthesis and degradation is going to be tightly regulated. So glycogen does have these high molecular weight granules for storage. So I know it can be a little bit confusing because we've got both dietary glycogen and the glycogen that we make. So just keep in mind that when we're saying glycogen, oftentimes we're going to be referring to the kind that our body makes, but we can go ahead and specify and say, oh yeah, there is also dietary glycogen. So if we're going to break down the glycogen that we make, so this is our storage glycogen. So storage glycogen is the kind that we make and we store in our usually muscles and liver. In order to do this breakdown, there's an enzyme called glycogen phosphorylase, and this cleaves a single sugar unit from the end of the glycogen chain. And it uses inorganic phosphate to phosphorylate the glucose. So notice this is drastically different from amylase, which amylase is going to work on dietary forms and just in kind of a somewhat random way of either disaccharides or trisaccharides or different things like that. Here, glycogen phosphorylase cleaves a single unit and phosphorylates it at the same time. When it does that, because it's phosphorylating it using inorganic phosphate, this is something that's going to be able to go into glycolysis, and it does not use an ATP molecule. So it is phosphorylating that single unit, but with inorganic phosphate, so no ATP required. Those limit dextrins are still going to be degraded by the debranching enzyme. So this glycogen phosphorylase is still going to have similar problems that the amylase has, and so it's still going to have a debranching enzyme to help even with storage glycogen. This glucose 1-phosphate can then be converted to glucose 6-phosphate by phosphoglucomutase. So remember that glucose 1-phosphate can't actually directly enter glycolysis. It's almost there though. So we're going to have one more enzyme, that phosphoglucomutase, to move that phosphate group from the first carbon to the sixth carbon. This reaction of removing one sugar unit and doing so with a phosphate group is called phosphorylase or phosphorylases. This glycosidic bond is split by phosphate, but it's not split by water. If it was split by water, there's another term we could use, which is hydrolysis. So realize that this term, phosphorylase, is very close because it's essentially the same, but instead of water, with hydro, we're not talking about phosphate, so it's got a phospho group for that. Also, if we were looking under standard conditions, this reaction would be very close to zero, but the delta G in vivo is going to be more exergonic because there's a high ratio of that inorganic phosphate. For muscles, that glucose 1-phosphate can undergo that one reaction with the phosphoglucomutase to enter glycolysis. In liver, you're still going to have that phosphoglucomutase going from glucose 1-phosphate to glucose 6-phosphate, and then that glucose 6-phosphate can actually enter that last step of gluconeogenesis to be hydrolyzed to glucose for transport to other tissues.